We know that spotty liver disease is a significant challenge in the industry at the moment. Before the start of this project, what was known about spotty liver disease and what are the gaps that this project is trying to figure out?
Spotty liver has been a problem in free range and barn production for quite a few years, and we knew it was bacterial because it responded to antibiotics, but we didn't really know what organism was causing it until a few years ago when the discovery of the organism was made. And scientists down at RMIT University worked with that and characterised the organism, named it, and proved that it caused the disease. And they named it Campylobacter hepaticus. So it took quite a few years to find that. Now it's been detected. We've got some tests developed that will enable us to find the organism in the environment or in the birds, and we can use that to augment the study that we're trying to do to find out factors that are associated with the disease in the field.
So is that the primary objective of the research, to work out the factors associated with spotty liver disease on farm?
Yeah, we're doing an epidemiologically based study. So we're going around a lot of farms, recording almost everything we can think of in the environment and management factors. So we're looking for factors that can modify the expression of the disease, whether either they provoke it or are protective against it.
So why is this research important for the lay industry?
Well, we need to know more about spotty liver disease because it's quite a troubling disease to the industry. It's defying a lot of understanding at the moment, and takes a lot of medication usually to overcome it in some situations. So if we can find why some farms don't get it and other farms do, if we can find some factors that contribute to either having it or not having it, we can help the industry find ways to minimise the effects of it.
So you've mentioned the outcomes of the project, but what will the final outputs be?
Well, hopefully we'll be able to come up with a list of do's and don'ts for spotty liver for free range and barn producers. There may be quite a complex lot of interactions between some factors, so we're going to have to try and identify which factors go together to create good or bad outcomes. So we're hoping to identify that sort of interactive relationship that'll help us control the disease. And we hope to come up with some recommendations on how to do that.
Could you just tell me a little bit more about how the project is being conducted?
Okay, it's called a case control epidemiological study. So it's a field survey. We've selected a group of farms, some of which don't usually see the disease and some which do. So we have our controls and our cases. And the concept is to measure everything we can on those farms and compare the two situations together so we can find things that may be associated with expression of spotty liver or prevention of spotty liver. So we're looking at pretty much everything, including rearing factors. So we're looking at things like density, litter types, perches, platforms, space allowances, what they're vaccinated for, some feeding and growth information, if they've been medicated, what their lighting programs are, if there've been any disease occurrences. Then we follow those birds through into the layer flocks, and again we're looking at density, perches, platforms, slat types, litter types, how good their production's been, their body weights, their health, what their lighting programs are, how many floor eggs they've got, whether there's been feather pecking, nest types and spaces, just drinkers and feeder allowances, the weather, and factors out on the range. So quite a large variety of things we're trying to cover and see if we can find things that tie together with the disease or not the disease.
Are there any results so far, or anything that you found that's interesting to date?
It's a little bit early for us, but we've had some surprises. All our rearing flocks that we've studied have been negative on cloacal swabs for Campylobacter hepaticus, although we did find it in one flock in feces in the rearing shed, and that's in a farm that quite often has spotty liver in the production phase. We have, as we said, flocks that don't get spotty liver and flocks that do, but nearly everything is positive for Campylobacter hepaticus. The organism is there in almost every sheds we looked at, except for maybe 2 in the cloacal swabs. So the organism's there, but we may or may not have the actual disease expressed. Farms we expected to be negative with no history of spotty liver actually had outbreaks during the survey, which was also interesting. We had one farm that had never recorded a case of clinical spotty liver disease. However, they did have not great production, but no mortality at the time, and the birds were 100% positive for Campylobacter hepaticus on their cloacal swabs. And the consulting veterinarian on that farm instituted an antibiotic treatment and actually got an improvement in production. So we're wondering whether that was a subclinical spotty liver case that was just sitting there without showing up as it normally does with mortalities. We've been to some farms where you had a number of sheds, and some have a history of having spotty liver, and others on the farm have never seen spotty liver. But when we checked the birds, they're all positive for Campylobacter hepaticus. So there's something interesting going on in between those two shed types that we're trying to determine. We also saw a brand new site, never had birds on it before, in an operation that had never seen clinical spotty liver before, and they broke in their first batch. So that was quite a surprise, and we don't know where that came from yet, but we're having a good look. The other interesting thing is we see it in barn sheds, and we've also seen it in free-range flocks before they've been let outside. So this does not seem to be necessarily related to ranging. It's a problem in the shed, which is something we haven't really focused on before. So now we're analysing across the occurrence of those sort of factors, and we'll see if we can identify some differences between those farms that are clinically affected and those ones that aren't.